Hello. Sorry I'm late. I'm not feeling real well.
Oh,
And
okay.
yep. All right, now we're recording. Thank you for catching that.
No problem.
All right, I want to briefly talk about special stains, mainly the capsule stain. It's used to stain the cells to look for capsules. Excuse me. When you stain with the capsule stain, it stains the background of the slide and it stains the cells, but then the capsules are not stained. Try me to blow this up a little bit. And so this looks clear or white where the capsule is. You need to know this because this is India ink, a negative stain, which stains the background of the cell, and it stains the cell very lightly. And then the capsule isn't stained. I want to talk about this stain, the capsule stain, because you may need this stain to perform your unknown test. Some of you will, some of you won't. Any questions about any of that? All right, let's begin chapter 4. So as always, I give an introductory slide giving you the major goals and the rough outline. I'm going to say, you guys know the terms. I'm not going to read through it because my throat is sore. And then we'll talk about, the last thing we'll talk about is the endosymbiotic theory in this chapter. And we'll also talk about how gram-positive and gram-negative cell walls differ and describe the importance of taxis in bacteria. So when we talk about prokaryotes and eukaryotes, prokaryote is Greek for pre-nucleus. And if you really want to translate correctly, it'd be pre-kernel, but the kernel is the nucleus. And then the eukaryotes is Greek for true nucleus. As you know, eukaryotes have a nucleus. There are differences in the DNA between prokaryotes and eukaryotes. Prokaryotes generally have one circular chromosome, and it's not in the nucleus, meaning the chromosome is simply in the cytoplasm. By circular, we mean that the ending point of DNA is the same as the starting point. So the DNA is in a circle. Eukaryotes have linear chromosomes, meaning they have the start position. Think about the X chromosome, the start position, and then an end position. And their chromosomes are in the nucleus. Usually with eukaryotes, there are numerous chromosomes. Their DNA is different in that the DNA of at least bacteria are not associated with histone proteins. The DNA of eukaryotes is wrapped around histone proteins, and histones are a protein, and that's all you need to know at this point. Prokaryotes do not have any membrane-bound organelles like the mitochondria, the Golgi apparatus, etc. The eukaryotes have membrane-bound organelles. Bacteria, and this is a bacteria only, meaning not the archaeobacteria, have peptidoglycan in their cell walls. So if the bacteria has a cell wall, we always find peptidoglycan in it. There is no peptidoglycan in the cell walls of eukaryotes. And of course, animals don't even have a cell wall. Prokaryotes divide by binary fission, meaning literally the cell splits down the middle into two. Eukaryotes, they divide by mitosis. Any question about any of that? All right, so we're going to talk about prokaryotes first, talking about cell shape and arrangement, the cell structures you can find in prokaryotes, the movement of materials across the cell membrane, and then lastly, we'll talk about eukaryotes. And I'll go through this very briefly because you should have had this in the introductory class. We'll talk about cell structures of eukaryotes, and then we'll end talking about the endosymbiotic theory. Prokaryotes have a specific cell size and a shape. They range on their cell size. Most of them are 1 to 2 micrometers long, but they can be smaller, as small as 0.4 or 0.4 micrometers, and they can range as large as 10 micrometers. The basic prokaryotic shapes is they can be round, and we call these coccus. They could be a rod, we call these bacillus, or they can be a spiral. We call those spirilla. Any question about that? Now, they can have a few other shapes, but most of them are a derivative of one of these three. Like you can have a vibrio, which is a curved rod, and then you can have a spiral, and then there's one with many spirals, and we call this a spirochete. These cells are a little different, and that is a spirillum. The cell wall is fairly rigid, whereas the spirochete, the cell wall is flexible. And I don't know, it gives this cell its spiral shape. But the cell, for example, can bend this one. It can be this way, and then it can come up that way, meaning this thing is really flexible. Any questions about any of that? There are some unusual shapes, like there's a star-shaped Stella bacteria, and then there's a square halo arcula archaea bacteria, which that actually looks a little more rectangular to me than square, but these are very rare, the unusual shapes of bacteria. Most prokaryotes are coccus, bacillus, or spirals. Most bacteria species are monomorphic, meaning that the cells of one species all have a single shape. Like these, they're all squarish. But a few species are pleomorphic, in which case they have many shapes. And this is a picture of a cell that has many shapes. The bacteria that tend to be many shapes are bacteria that do not have a cell wall. And we'll briefly discuss that later in the term. And like I said, the shapes of these cells are pleomorphic. When we talk about the cocci, the cells can be individual coccus, or they can be diplococci, where two cocci are stuck together, or multiple cocci stuck together in a row, and that we call streptococci. You can also have a tetrad, Sarxinia, and that's 8 cells stuck together. And then the last would be staphylococci. And this literally means a cluster of grapes, because when they saw that, they reminded of a cluster of grapes. The reason for what the cellular growth arrangement can be is how the cell is dividing, meaning its plane of division, and whether the cell walls stick together. Now, I should warn you that whenever you have one cellular growth arrangement, like streptococci, you will see streptococci, but you'll also see some diplococci. And what happened was this cell broke, or the streptococci broke into diplococci. And then you'll also have single cocci, meaning this broke apart here. So the cellular growth arrangement is always pleomorphic. And when you're trying to determine the cellular growth arrangement, you find the highest cellular growth arrangement and say that that's what it is, streptococci. Now, when I give you the gram stain for your unknown, we're going to be taking cells from a petri dish and then staining them for the gram stain. And because of that, the cells stick together, in which case you cannot determine the cellular growth arrangement. And I'll try and warn you of that earlier, meaning the cells stick together. So instead of being streptococci, which I already told you with streptococci, you'll see diplococci and single cocci, they stick together. So you might see some tetrads, some sarxinia, and even some staphylococci. And to get the cellular growth arrangement, what you want to do is grow the culture in a liquid culture, meaning a broth. And then the cells don't stick together. And then you can more accurately determine the cellular growth arrangement. All right, any question about that? Now, the cocci have the most cellular growth arrangements. The bacilli have some cellular growth arrangements, but they have fewer. You can have single bacillus, you can have diplobacillus, blow this up a little. You can have streptobacilli, and this is where they're hooked together on the long end. When they're hooked together on the short end, we call this palisade. And if you remember reading Mark Twain, he talked about a palisade fence. And if you stand this on this end here, and this end going up, what it is it's simply a board, and then a board, and then a board, and that's the palisade fence. And that's what they named this growth arrangement after. This is not a growth arrangement. This is a different cell shape. And that is when you get a bacillus that's rounded on the edge, and then we shorten it and shorten it, it will no longer look like a bacillus. It'll look between a coccus and a bacillus. And this we call a coccobacillus. And here are some coccobacillus. Any questions about any of this? Once again, with the bacilli, you will not see only one cellular growth arrangement, even when you grow it in a liquid culture. If it's streptobacilli, sometimes this will break and you'll see diplobacilli. And then this can break and you'll see single bacillus. So the cellular growth arrangement is always pleomorphic. Any question about that?
Are these different shapes just like a way to describe the cell, or are they like a real genus name or a real species name?
It is a cellular shape, meaning there are bacteria that take on the bacillus shape, and there are bacteria that have the cocci shape. But you should realize that the word bacillus does have two meanings. If it's spelled with a lower B, lowercase b, we're talking about the shape of the cell. If it's with a capital B, that is a genus, the genus Bacillus. Okay,
Everything in the genus Bacillus is rod shaped, right?
right. And then there's a Bacillus, a genus Streptococcus. And it is cells with streptococci.
But what about for like the other ones, the pair, the 4, the 8, and the cluster?
Yeah, there's no genus with those names. And then streptococci is slightly different than streptococcus. Okay, so the genus will be a capital S and it'll be streptococcus. The cellular shape will be a lowercase s and we call it streptococci. All right, so let's talk about some parts of the bacteria cell. All bacteria, and actually all cells, have the structure shown in red. Cytoplasm, ribosomes, plasma, membrane, and bacteria have a nucleoid region. In eukaryotes, we have the nucleus. The nucleoid region is just wherever in the cytoplasm the DNA is floating around. And it stains usually a little differently. So right there, we're seeing the nucleoid region. It stains a little differently than the rest of the cytoplasm. But there is no boundary between the nucleoid region and the cytoplasm. The nucleoid region is wherever the DNA floats in the cytoplasm. Any question about any of that? There are some other parts to bacteria that some cells have, but not all bacteria have, like the capsule, a plasmid, a pilus, inclusions, a capsule, a cell wall. Most bacteria do have a cell wall, but there is one genus that does not have a cell wall. I'm not sure why that isn't red. It's red over here. The plasma membrane is in all cells. And then Fimbriae and flagella. These are things that you see in some cells. Excuse me. The glycocalyx is secreted by many prokaryotes, but certainly not all prokaryotes, and probably less than half of prokaryotes have a glycocalyx. It's outside the cell wall, it's secreted, it's usually sticky, and there are 3 different types of a glycocalyx. The capsule is neatly organized glycocalyx. It prevents phagocytosis from white blood cells. And the capsule is what you can see under the light microscope. So if you can see the glycocalyx under the light microscope, it is a capsule. The slime layer is unorganized glycocalyx. It's mainly water and it's fairly loose. Because it's mainly water, you can't see the slime layer under the light microscope. You would need to look at with the electron microscope to find the slime layer. And then there's a third glycocalyx, and that's called the extracellular polysaccharide. You can find the extracellular polysaccharide in cells that are secreting a biofilm. And it's the extracellular polysaccharide that makes the biofilm. And we will talk a little bit about biofilms later in the term. All right, any questions about any of that? So glycocalyx is all three of them. The one you can see under the light microscope is a capsule. Flagella is used by the cell for locomotion. And so if the cell can swim in a liquid culture or a culture that is similar to a liquid culture, bacteria have a flagella because that's the only way that they can move. The flagella is largely outside the cell wall. Meaning here's the flagella here outside the cell wall, but there's a small portion of it. Here's the cell wall right here. There's a small portion of it that goes through the cell wall. And there's even a portion of it that's hooked to the plasma membrane. So it's mostly outside the cell wall, but it's not entirely outside the cell wall. The flagella is made of proteins, which we call flagellin. And remember, if the bacteria can move, it has flagella. You can have different bacterial arrangements, and you don't really need to know the terms. I won't quiz you on this, but the flagella can be on one side, or it can be on both sides. It can be multiple on both sides, or the flagella can be all over the place, in which case we call it peritrichous. Motile cells have flagella, and the cells can rotate or swim. And it looks like the cells are running or tumbling when you look at them under the microscope. And this can give the cell taxis, movement toward or away from a stimuli. So taxis is movement toward or away from a stimuli. We have two different types of taxis: chemotaxis, and that's movement toward or away from a chemical stimuli. And we have phototaxis, movement toward or away from light. This one here is phototaxis, and it looks like it's moving away from the light because the flagella are on this side. So that means the bacteria are moving that way. The light is this way. So it's movement away from the light. That's sort of important for photosynthetic organisms. They usually have phototaxis, and they're attracted towards the light. And so they'll come up to the surface. But that cell obviously isn't photosynthetic, and so it was moving away from the light. A structure that we find in some cells is called the axial filament or the endoflagella. It is a flagella, but it's inside the cell wall. And you only find it in spirochetes. So here, when we look at the cell, we can see that the flagella is wrapped around the cell. And if you look here, let me blow that up. If you look here, the axial filament or endoflagella is inside the cell wall. And there's staining of the axial filament seen under the electron microscope.
This is
I have a little video here. You can see how flexible the spirochetes are. And there you saw the cell just changed its direction. And it can move different ways by varying which way it is, I don't know, whatever you want to call it, the spiral movement. So if it's spiraling this way, it's moving that way. But if it will be let me see if I can get this done again.
This is your living room / yoga shanti / regional office / and this.
So the spirochete is moving that way with this spirilla movement. And now it's switching and moving that way and then that way. Anyways, the point is the spirochetes can be very flexible and they can vary their movement. This movement is believed that spirochetes can actually burrow through human tissue with that spiral-like movement. And certainly, like syphilis, it's thought that it can burrow through the intact mucous membrane by the spiral-like movement. All right, any questions about endoflagella, also called the axial filament? Fimbriae are shorter and straighter and thinner than flagella. So if the cell has a few and they're longer, then it's a flagella. And if the cell uses that to swim through liquid media. The fimbriae, the cell uses it for attaching to surfaces, meaning the fimbriae are not used for swimming. And some human pathogens have fimbriae, which the fimbriae makes the cell pathogenic because the fimbriae allows the cell to attach to our cells. The pili is also called the sex pili, and it's a little longer than fimbriae, and it's also hollow. It's used to transfer DNA from this cell into that cell, and we'll talk about sex pili later in the term. Only a few prokaryotes have sex pili. All right, the prokaryote cell wall is complex. It's a semi-rigid structure responsible for giving the cell its cell shape. It protects the cell and prevents osmotic lysis, so that if a bacteria were to fall into a beaker of water, for example, as long as it has an intact cell wall, water would move into the cell only up to the point where the cell membrane reaches the cell wall. And then the cell membrane can't expand any further because the cell wall is protecting the cell. The cell wall is located outside the cell membrane. It does contribute to the pathogenicity of some species, and it's mostly made of a peptidoglycan. We will talk a little bit about that later. The main molecule in bacteria inside the cell wall is the molecule of peptidoglycan. And the peptidoglycan is what gives the cell wall its semi-rigid structure. Peptidoglycan is a polymer of repeating disaccharides. And the two disaccharides are NAG and NAM. Let me blow this up. NAG is N-acetylglucosamine, which probably means as much to you as nag, and NAM is N-acetylmuramic acid. And the point is that I can't read this, so this looks like it's NAM, NAG, NAM, NAG, NAM, NAG. It just repeats one after the other. And then we have a row of that, and that's next to another row of that, next to another row of that, next to another row of that. And then underneath this plane, we have another plane composed of NAG and NAM polysaccharide linkages. And even in the gram-negative cell wall, we have multiple levels of polysaccharide, the peptidoglycan. Now it's called peptidoglycan because glycan is a sugar, meaning NAG and NAM are the sugars, but it has short peptides hooking the peptidoglycan together. And there's one peptidoglycan, one peptide linking this NAG and NAM with the one below it. And then this right here is another peptide which links this peptide with that peptide, meaning that this peptidoglycan linkage of NAG and NAM is hooked together with the peptide with this one. And that's why it's called peptidoglycan, because glycan is the two sugars, and then there are short peptides linking the sugars together. Any question about that? Now let's talk about the gram-negative cell wall. The gram-positive cell wall. When we talk about the gram-positive cell wall, the yellow is the cell membrane, and then the cell wall is thicker and it's purple. And when we look at it this way, there's the cell membrane, the lipid bilayer of the plasma membrane. And then the peptidoglycan is on multiple layers. And peptidoglycan is the main molecule in the cell wall of bacteria gram-positive cells. Now note that this peptidoglycan, let's see, we're seeing 3 different layers here. It's more than 3 different layers. It's probably hundreds or thousands of layers of peptidoglycan in the gram-positive bacteria cell wall. There are some other molecules in the cell wall, and we'll talk briefly about them. There's teichoic acid, and that just runs through the peptidoglycan, helping to link the peptidoglycan together. There's also lipoteichoic acid, and this runs through the cell wall as teichoic acid, but it has a lipid portion, and the lipid portion binds to the cell membrane. So the lipoteichoic acid binds the peptidoglycan to the cell membrane. And that's about it for the molecules that we see in gram-positive cells. Any question about any of that? In the cell wall, I mean. All right, I've already talked about that. The teichoic acid may help regulate the movement of cations, meaning a positively charged ion appears to like to move down the teichoic acid. However, the cation can dissolve in water, and the cell wall is water soluble, meaning water can go right through the cell wall. And so a cation can dissolve in the water and go right through the cell wall. But the teichoic acid does help regulate the movement of cations. The polysaccharides can provide antigenic variation. For example, E. coli, what is it? 0151, something like that, has polysaccharides which give it the antigenic variation. Now, the gram-negative cell wall is a little different. And when we look at it this way, we have a lipid layer, which is the cell membrane, internal to the cell wall. And then we have a peptidoglycan layer that's part of the cell wall. And then we have a lipid bilayer on the outside of the cell wall. This is called the outer membrane of the cell wall. When we look at this in greater detail, there's the lipid bilayer of the cell membrane. And then there's the peptidoglycan layer. Now this is showing you only one layer of peptidoglycan. In reality, there are many layers of peptidoglycan, but it is fewer than the layers in a gram-positive cell. And I mentioned in the gram-positive cell, you can have hundreds or thousands of layers of peptidoglycan. In the gram-negative cells, you have fewer layers, but it's more than one. Everybody got that? And we'll just say there are several layers of peptidoglycan, but probably fewer than 100. And then this is part of the cell wall, the peptidoglycan, and this is part of the cell wall also. And as you notice, this part of the cell wall is a lipid bilayer. This portion of the outer membrane of the cell wall is the same as the plasma membrane, meaning phospholipids are the main molecule in the lower layer of the outer membrane of the gram-negative cell. On this upper layer of the outer membrane of the gram-negative cell, we do have phospholipids. They're the yellow ones in the linkage here. But we have another lipid, and it's actually bound to an old polysaccharide, which gives it a hair. And that's the blue one here. So that's a lipopolysaccharide because the roundish ball-shaped object is the lipid A, and it's obviously a lipid. And then it's bound to an old polysaccharide, which gives it the hair. And so this molecule is a lipopolysaccharide. Now this molecule is extremely important because lipid A is an endotoxin. And so if a patient ever has a gram-negative infection, that means they're going to be exposed to lipid A, which is an endotoxin. And we'll talk more about toxins later in the term, but toxins are something which are toxic to us. And so if a patient has a gram-negative infection, they are exposed to an endotoxin. Any question about any of that? Now, lipid A, we will talk about 3 times in this class. And this is the first time we've talked about it. Any questions on that? Now, we do find a few other molecules in the cell wall of the gram-negative cell. We find proteins in the cell wall. And this one, as you notice, is going through the outer membrane of the cell wall. And it's also going through the peptidoglycan. I don't know if that's going through the cell membrane or not. I can't tell from this picture here. And I can't tell any of them, to tell you the truth, if they're going through the cell membrane. But the point is, this protein is spanning the cell wall. There's also another protein, which is called the porin protein. And the porin protein makes a channel in the outer membrane of the gram-negative cell wall. If the cell wants a molecule to come into the cell, like glucose and an amino acid, this outer membrane would keep, the lipid bilayer of the outer membrane would keep those molecules from getting through. But the porin protein provides a channel to allow those molecules to move through the outer membrane of the cell wall. And then those molecules are water soluble, so they can move right through the peptidoglycan. And then they have to move through the cell membrane. And to move through a cell membrane, we have a transport protein, which generally moves or transports one molecule across the cell membrane. So we have a transport protein, for example, to move glucose into the cell. We'd have a transport protein to move fructose into the cell. We'd have a transport protein to move methionine into the cell. And we'll have one different protein to move all of those different molecules through the cell membrane. Any question about any of that? So the movement of the molecule through the cell membrane is very specific. One transport protein only moves one molecule through the cell membrane. The porin protein moves many different molecules through the outer membrane of the cell wall. Any question about any of that? I think I've already talked about this. So the gram-negative outer membrane has a few different molecules in it, including the lipopolysaccharide, lipoproteins, phospholipids, and there's a periplasm, which is the space between the outer membrane and the plasma membrane. And the periplasm is where you find the peptidoglycan. The gram-negative outer membrane does give the cell protection from lysis, like if the cell falls into a beaker of water. So it does protect the cell as the cell wall does. But the outer membrane of the cell wall can also protect the cell from complement proteins and from antibiotics. The complement protein is something that our body makes to attack foreign invaders, and we might talk about that later in the term. And one example of antibiotics is penicillin G. It cannot move through the outer membrane of the gram-negative cell wall. So penicillin G will not be effective on gram-negative cells. I mentioned this earlier, and that is the gram-negative outer membrane can have the old polysaccharide, which can give antigens for the cell, like E. coli, O157H7. The old polysaccharide is the O157, which gives the E. coli different antigens. And then I mentioned lipid A is an endotoxin. So if the patient ever is exposed to gram-negative cells with the cell wall, you're going to be exposed to an endotoxin. And then the gram-negative outer membrane has porin proteins that allow molecules to enter and leave the outer membrane of the cell wall. I think I've already talked about this. Let's move on. I think I went backwards for some reason. So there are some differences between the gram-positive cell wall and the gram-negative cell wall. The gram-positive cell wall is a thick peptidoglycan. It's a very rigid structure. The gram-negative cell wall is thin peptidoglycan, and it's fairly rigid. It's not as rigid as the gram-positive cell wall. We can find teichoic acid in the gram-positive cell wall. There's no teichoic acid in the gram-negative cell wall. There's no outer membrane in the gram-positive cell wall, and there is an outer membrane containing lipid A in the gram-negative cell wall. The cell wall does allow the gram stain to differentially stain the cells, meaning the crystal violet iodine complex gets stuck in the thick peptidoglycan of a gram-positive cell wall, and they do not leave the cell during the acetone wash or the decolorizing step. The gram-negative cell wall has a thinner peptidoglycan layer, and the alcohol dissolves the outer membrane because alcohol dissolves lipids. And then the crystal violet iodine complex washes out of the thin peptidoglycan. The cells stain pink because we then apply the cancer stain, safranin, which stains the cells pink. Any question about any of that? And this slide is showing you some differences between the gram-positive cell and the gram-negative cell. And you only need to know the top things. You can ignore the bottom things. I'll let you read that because I'm not feeling too well. There are some atypical cell walls. Let's talk about the bacteria first. The mycoplasmas are cells which lack a cell wall. They typically have sterols in their plasma membrane, which is similar to us, meaning animals, typically have sterols like cholesterol in their plasma membrane. And the cholesterol in our plasma membrane helps give strength to the plasma membrane. Well, the mycoplasmas don't have cholesterol, but they have another sterol in the plasma membrane, and it does the same. It helps give strength to the plasma membrane. Now, the mycoplasmas, when you stain them with the gram stain, it stains gram negatively. Why does it stain gram negative? Nobody's going to guess. Okay, it has no cell wall, so the crystal violet iodine complex washes out of these cells, and then the cells stain with the safranin, so the mycoplasmas stain as a gram-negative cell. But recall that they have no cell walls, and they are a genus of bacteria. And they usually are pleomorphic because they take on different shapes because they have no cell wall, which gives the cell its structure, its shape. The mycobacterium are another family of bacteria, and their cell wall is a little different. They have a lot of lipid in their cell wall, and the lipid is called mycolic acid. And we call these acid fast staining cells because the acid fast stain gets in the mycolic acid and stays there. We will take a look at the mycobacterium later, and I don't think I have a picture of it here. We'll talk about the mycobacterium later in the term. They are gram-positive cells. But they're a little different. Let's come to here. And that is they are gram-positive like this, but in the cell wall, they have mycolic acid. And they have a fair amount of mycolic acid in the cell wall. And that gives those cells different properties. For one thing, the mycolic acid can prevent water-soluble substances from coming into the cell or leaving the cell. And it's thought that the mycobacterium, the cells can live a long time under desiccated conditions because the mycolic acid prevents water from leaving the cell. All right, we'll talk about the mycobacterium a little bit later in the term. And then there's one other atypical cell wall, and these are not bacteria. They're the archaea. All of the stuff that I talked about with the cell wall is the cell wall of a bacteria. The archaea have different cell walls. Instead of having peptidoglycan, they have pseudomurine, which is another disaccharide. And some of the archaea actually even lack a cell wall. So the archaea have different cell walls, okay? And we're not really going to talk much about the archaea in this class. And the reason for that is not that they're interesting, it's just that there's no archaea known that causes human disease. All right, I think I'm going to stop here. So are there any questions? And I will see you at 2:30 for the lab.
I have a question.
Yeah.
So what exactly is going to be on tomorrow's quiz? Chapter 2 and then 3?
Let me take a look at the syllabus. And you should note that I never changed the quiz. I meant to do so, but I've been feeling so lousy, I haven't changed the quiz. I haven't even read what people made a comment, but the greatest comment I saw was that people were willing to take the quiz from something like 8 p.m. to 8 p.m. So 8 p.m. on Tuesday to 8 p.m. on Wednesday. I'm not feeling very well, so I doubt that I will get that set up for the next quiz. And if that's a problem for one of the students, contact me and I'll try and make an exception again. And I'm sorry, I just haven't been feeling well. But the quiz for Wednesday is covering labs 3 and 4, chapter 3, and the end of chapter 2. Okay?
Okay.
Dr. Volpe, I have another question.
Uh-huh.
I'm the student with the weird schedule, so I was just hoping if you could make that exception for me tonight, that would be super helpful.
Uh-huh. Yeah, talk to me in the lab and I'll try and get that done, okay? It
Thank
might
you.
be I could even make all of them done tonight. We'll see. The trouble is I hate changing it so early, but because there's very little time.
Yeah,
Yeah.
I appreciate
so
it.
I'll probably make an exception for you, all right? Just talk to me in the lab, all right?
Okay.
All right. All right. I'll see you in the lab.